Okay, perfect. We can see it clearly.
Is that
Let's
good?
just see if we can hear. Yeah, we can see it and we can hear you loud and clear as well. So I'll just remove myself from the video and mute myself. Then the floor is yours. Johnny, the floor is yours.
Thank you very much, Ruben. Good afternoon, everyone. And thanks to invite me to present this lecture. It's great to see from those planned lectures the incredible diversity that is on offer. And I hope to do today is to both give you some high-level probing questions and thought-provoking ideas, as well as giving you some of our sort of more detailed approaches to the problem. So, I've now spent quite a long time learning and teaching immunology to different groups of medical students and scientists. And for the most part, people find immunology very daunting. And unfortunately, it is complex, but I think it is the complexity and our modern technologies that are allowing us increasingly to get to the level of precision that we need to start to understand the immune system, as well as to start to manipulate it effectively. So for many, many millennia, really, the human immune system has been evolving alongside different pressures from the environment. And really, if you think about infectious diseases, we've recently faced the pandemic of a new RNA virus. But for millennia before that, if you look across mycobacterial, DNA viruses, infectious parasites like malaria, these have been shaping the human immune system. And by doing that, it's largely because the adaptive immune system is designed in such a way to have a huge diversity in its repertoire. And that is confined across different populations. And really the whole point of that biological diversity is to ensure that if any infection arises or any problem arises, there will be a group of us that will be able to effectively manage and survive that onslaught. And in so doing, through the millennia, infectious diseases has shaped the way our immune system sees the world. Much more recently, though, has been the introduction of drugs. And so where infections were experiments of nature, we can really look at drugs and also increasingly other small molecules in the environment as new iatrogenic experiments of humankind that also probe our immune system. So if you look at adverse drug reactions, whenever we introduce a new drug for a therapeutic effect, there are invariably adverse drug reactions that occur. And these are drug reactions that can either be on target, so that's where the therapeutic action of the drug will result in a problem. And so here I put on this slide an example: when you give somebody an antibiotic, you will on target destroy commensal organisms in their gut, and that comes sometimes with a pathological consequence, such as an overgrowth of the pathological organism Clostridium difficile. In addition, though, there are lots of what we call off-target adverse drug reactions. And the important thing about this is that not all of those are mediated by the immune system. So lots of drug targets include other cellular proteins, both on the membrane and intracellular, and can cause different insults to cells, and those can result in different toxicities. And here I put one severe toxicity related to aminoglycosides giving damage to the kidneys. This is not mediated at all by the immune system. What I'm going to talk about and focus on today, and what I'm interested in, are those drug reactions that are mediated by the immune system. And they come in a wide variety of phenotypes, and also in terms of timing. So they can have a quick onset. People in this audience, maybe the general public's often familiar with something like anaphylaxis. The person takes a drug, and within minutes, or even sometimes even a couple of seconds, some patients have a reaction. This can be mediated both through traditional antibody-mediated mechanisms, which I've shown in section 2 there, or it can be, recently it's described that it can work through direct receptors on mast cells, which lead to mast cell degranulation. What I'm going to probe a little bit further in terms of our TB drug reactions this afternoon are those right on the end here, which are mediated by interactions between drugs and T cells. So when one considers really from basics, building up and thinking about conceptually a drug hypersensitivity reaction, there are a couple of key questions that one needs to say. The first thing, like I mentioned in the slide before, is in fact the mediator of pathology and immune cell and immune molecule. So is this actually an immunological reaction that you're seeing? Then the second part about it is, what are the main immune cells involved and what are the main molecules, either excreted molecules or actual cell-to-cell contact molecules that are causing the tissue damage and problem. And then digging down to the details is really this third question, which is that how and where does the drug actually interact with the immune system? And I'm going to show you in the context of small molecule drugs, how these can interact with the adaptive immune system. And then a question that goes way beyond drugs in general and looks across other allergens and other interacting molecules with the immune system is, why do some people develop hypersensitivity or allergic reactions and others develop immune tolerance? And I think that that is a broad question, and drugs offer us some insights into this. For those of you that are not clinicians, I'm going to really focus in on two main kind of what we call extreme phenotypes. And these are severe drug reactions that occur that affect the skin. So the first one we're going to talk about that's classified as severe cutaneous adverse drug reaction is something called Stevens-Johnson syndrome or toxic epidermal necrolysis. And what happens in this condition is there is an immune targeting of the keratinocyte cells that sit in the epidermal-dermal junction of your skin. And what happens is during that attack, you effectively get sloughing or damage and death of the epidermis and sloughing of that off the dermal layer of the skin. This affects, it can affect the entire body and can affect all mucosal surfaces. And you can see here in this picture on C, you can be left with this exposed dermis, and that can have serious life-threatening complications in terms of exposure to dehydration, inability to control fluids and renal failure, organ dysfunction, sepsis, and in many instances around the world, in older cohorts, particularly the mortality rates can be 60%. Similarly, also with epidermal necrolysis, but with a more confined area, something we call bullous fixed drug eruptions. Fixed drug eruptions are these very circular, well-demarcated patches that have inflammation and then subsequently heal with pigmentation. In the severe form of this condition, you get actual blistering and loss of the epidermis. And then the other group of reactions that I'm going to talk about, also what we call a severe cutaneous reaction, is something we refer to as DRESS syndrome. And that stands for drug reaction with eosinophilia and systemic symptoms. And what happens there is really it's a immune-mediated reaction that gives rise to this diffuse erythematous infiltrative rash. So you'll see, I'll show you some histology with infiltrating immune cells into the skin. Similarly, you also get abnormalities, particularly of liver. And so you get liver enzyme derangements. And then accompanying this reaction is actually what we call an eosinophil. So one of the immune markers in the blood is elevated. And this is these combination of symptoms and pathologies we term together in this DRESS syndrome. And these are really the focus of what we're going to talk about today. The other important context really for us studying this relates, unfortunately and sadly, to our burden of disease. Because if you look at drugs and their exposures in different populations, what drives their ability to probe the immune system is really prescribing habits connected with the diseases that underlie them that they are used for treating. And unfortunately, in South Africa, we have these dual burdens of TB and HIV. And particularly, I call out this point at the bottom here, which is that at the moment, we still see about 60% of incident TB cases occurring amongst HIV co-infections. So often we see both HIV, TB, and then we see the two together. And of relevance this has to drug hypersensitivity is really on this slide, which is that HIV in South Africa is responsible, together with TB, for the greatest burden of these severe cutaneous adverse drug reactions, also responsible for the greatest burden of other organ-specific drug immune reactions, such as drug-induced liver injury. And although the figure is not clear, the estimated increased risk is up to 100-fold amongst HIV infected patients for these immune-mediated reactions. Fortunately, though, the majority of these reactions, especially the ones involving the skin, are actually mild. So just for instance, a mild rash. And usually that can be treated, the drug can be continued through that rash, and the person gets immune tolerance and does not get a severe reaction. Similarly, if you look at the drugs causing the more severe reactions, you'll see the commonest drugs being antimicrobacterials or anti-TB agents and other antibiotics like cotrimoxazole, which we use for prophylaxis against pneumocystis in severely immunosuppressed patients. And if you look across the condition of epidermal necrolysis, which the epidemiology of which is highlighted on this global map, what you see is that this occurs throughout the world, but in different populations, the drugs causing this are quite different. So you see here commonly the anticonvulsant drugs, you see some non-steroidal drugs, allopurinol is a common offending agent. And then in the context of an HIV setting, we see a Bacava-related hypersensitivity. And then in our continent, based on the slide I showed you previously, the bulk of the burden is amongst first-line TB drugs and also Bactrim. And here you can see the amount of cases that we see. We see about up to 45 of these new cases in just our single tertiary center at Groote Schuur Hospital alone. And to give you some idea, not just of the mortality of this condition, but the morbidity associated with this condition, it costs these patients, these patients often spend several weeks to months in the hospital in terms of managing them back to health. They're often left with considerable post-reaction morbidity, particularly affecting the genital areas, affecting the nails and the teeth, reflecting the eyes. And this can be a cause of major long-term morbidity. And then just in terms of the cost to the health system, these rack up into the thousands of dollars in order to safely care for these patients. If you look across stress, which is the other severe cutaneous reaction that I mentioned, you can see here a very different pattern in South Africa compared to what we see globally, where there is also other antibiotics, but also, again, drugs like allopurinol and the aromatic anticonvulsants. Since 2016, we have formed a multidisciplinary drug allergy clinic, which brings together subspecialists in dermatology, allergy, pharmacology, infectious diseases, nephrology, and hepatology. And it brings it together, and we have come together to really care for these complex patients. And stemming from that clinic, we have established a registry and a biorepository for looking and studying these cases longitudinally. And furthermore, we have formed a network across other sub-Saharan African countries, which you can see here termed AfriSCAf. And this just gives you some idea, just within the first 3 years of our registry and repository, what we have managed to collect. You can see here the predominant phenotype is that of DRESS, but also we see a fair amount of SJS-TEN. The commonest offending drugs are the first-line TB drugs, and then second, Bactrim. And then you can see we have very little of the other drugs that affect lots of other places globally. I think that a lot of the starting insights that we have around these conditions and what it means about the interactions between drug and the immune system have really stemmed from the clinical conditions that we have been faced with managing. And in the slides that I'm going to show you next, I really want to call out the work of my close collaborators at the Drug Allergy Clinic, Professor Leguenya, who is the head of dermatology, and Professor Dlamini, who is from the infectious diseases department. The two of them, long before I actually returned to look more at the mechanistic side of things, have been conducting and pioneering much of this clinical work in the care of this challenging group of patients. But what are some of the key challenges that we are faced with? Well, this is the typical patient that presents with DRESS. Firstly, the patient has advanced immune suppression. They have TB, which is often disseminated. And those two conditions together carry often a 6-month mortality of around 25 to 30%. So in other words, this is a very sick group of patients. The other problem that's faced is that at the time that they present with these reactions, patients are often on 8 or more drugs. So determining which is the offending agent is a particular challenge. They often have the recent introduction of antiretroviral therapy, adding more drugs and also the problems around immune reconstitution. And then, of course, I've already mentioned to you the severity, both from a life-threatening perspective and also from the morbidity of having one of these severe immune-mediated reactions. So really what that does is that creates the problem of the patient presenting with severe drug hypersensitivity on the one hand, and yet having an urgent need for effective antimicrobial therapy for a life-threatening combination of TB and HIV. And if you look at what options you have in that situation, well, the one is a decision to be made that this is not a severe reaction you can treat through. But of course, I've shown you that these patients I'm talking about are the ones with the severe reactions and not those mild group that can be treated through. You could opt to give the person no treatment, but unfortunately, that would lead to death and a very high mortality from the actual infectious diseases. You could look at alternative drugs, and this is certainly an area of a lot of development in the context of TB, with different and newer TB agents for TB being developed and different short course regimens. But unfortunately, still, first-line TB drugs that we are studying, particularly the rifamycins, remain pivotal microbactericidal drugs that are cost-effective for use in low middle-income countries. And so it doesn't look like anytime soon we will be able to find a good alternative regimen for that. And of course, the other challenge with TB is if you give it exposure to just one or two drugs, you quickly develop resistance. And so it's critical that you have multiple drugs for prolonged therapies. And that also adds another element of challenge. What you could do is drug exposure and drug challenge, or you could think in some rare instances of desensitization. I would make the comment that in the context globally of these severe cutaneous adverse drug reactions, pretty much drug challenge and desensitization approaches are completely contraindicated, and people would like to avoid at all costs future re-exposure to drug. However, in our situation, that has not been the case, which has called for some innovative and collaborative approaches. So we put together now and have got a lot of the clinical experience in several aspects of dealing with these conditions. The first is making sure that when we look at after these patients, we really understand the clinical extent and the severity of them, and also how, if we give them supportive care and management, for instance, with topical steroids, what are their outcomes? And I'll show you in a subsequent slide how we actually discovered that our group of younger HIV patients with epidermal necrolysis have much lower than expected mortalities compared to other places around the world. And so that's the one aspect of it. Then we've also become really good detectives in filling out these chronologies where we carefully try to determine which drugs people have had introduced when, because these reactions have very clear latencies. In other words, they have very typical periods between when the drug is reintroduced and when the drug reaction develops. And often you can use that to give you a very good idea of what the offending drug might be. And then we've had to develop some novel approaches to actually considering drug re-challenge in the context of these reactions. But part of that was to initially go to basic allergy approaches in the context of drug hypersensitivity. And you can see several years ago, this is what Professor Luquenya and colleagues did, where they tried to do skin prick testing and patch testing for these patients to TB drugs. And here you can see an example of patch testing and skin prick testing being done with rifampicin that has this characteristic orange color. And you can see that being placed on patients' back. What they found, unfortunately, though, was that the sensitivity, the likelihood of picking up a positive reaction to one of the TB drugs was very poor, especially in HIV-infected patients. And then, in fact, as a surprising consequence, what they noted as well is that of the patients that were positive to these patch or skin prick testing that were also co-infected with HIV, almost all of them developed not just the localized reaction, which we would hope for around the patch, but actually a systemic reaction. Often this was spiking a fever or having worsening of their reactions. And this was in contrast to the bottom figure here, which showed that in HIV uninfected patients, the sensitivity was also low, but at least patients tended not to develop these systemic reactions. What that actually did, given the necessity and severity of the infection and this experience with these epicutaneous testing, was it created a sort of a risk benefit that favored potentially thinking about oral re-challenge or drug reintroduction protocols in this context. And as a result, Prof. Luquenya has really pioneered this what we call sequential additive drug rechallenge process. And you can see here that what happens is the patient is admitted to the ward. Once their clinical and laboratory features normalize, they then are sequentially re-exposed, first to isoniazid, then to rifampicin, then pyrazinamide, then ethambutol. And each drug is reintroduced with a 4-day time window. And if there's any symptoms that develop, a worsening of the rash, a fever, laboratory abnormalities, that drug is then considered to be one of the culprit offending agents, is stopped, and then the other drugs are introduced. Ranks also examined whether or not we should do graded or full dose drug provocation testing and found that full dose was safe and effective. And then what we do see is we are constantly trying to improve on this because we are aware still that exposing patients orally to drugs that they might react to in this context may be dangerous. And so we are looking, and I'll show you ways in which we've tried to optimize some in vitro diagnostics for these patients. And sometimes we will individualize these approaches. What we've seen now, and this is some of our unpublished data that we have hope to soon publish, looking at, these are probably the largest group of SCAR patients with drug provocation testing in the world. And what you see here is that the reactions they develop are quite diverse, ranging from skin symptoms with itch and rash, systemic fever, and sometimes even GIT disturbance and laboratory abnormalities. But the important thing to note is really on the side is that none of these reactions that we've had have been considered to be life-threatening through these severity criteria that we use for adverse drug reactions. Furthermore, we have found a method whereby if immediately on the development of the reactions, and you can see I've highlighted this red box here, on 6 patients where within the first hour to 2 hours, we've been able, on them developing a positive reaction to one of the drugs, treat them with intravenous methylprednisone, usually the dose of 100 to 200 milligrams. And what we've been able to do with that is abort these reactions. And now we are putting together, we now have almost around 30 of these patients that we've treated in this May with similar very good results. And what this is doing from an international perspective is helping people in some, especially some of the milder phenotypes of these severe cutaneous reactions, to consider in other drugs that potentially the benefit of identifying the offending drug in a safe manner is actually potentially the preferred clinical approach to managing these patients. But what about the immunological side? Well, what we've noticed, interestingly, is that there are different types of sequential drug rechallenge reactions. So in some patients, and you can see from these pictograms here in A, in some patients, they develop you reintroduce the drug and they develop a fever and some mild itchiness, but actually you can continue treating them with the drug and they will eventually stop having symptoms and you'll be able to safely discharge them on the drug. In others, like in B, what you see is following this initial positive reaction, other markers begin to vary and worsen. Usually the infiltration of the skin or the liver abnormalities will worsen. And those patients are the immunologically mediated hypersensitivity patients that cannot be tolerized. And those patients are another group. And then we have quite a few patients where either the reaction develops sometimes later, or for instance, the immediate reaction is worrying to the clinician and the patient is stopped. And that's a sort of indeterminate group that we are still trying to understand a bit better. I've showed you the differences in clinical phenotypes because the reactions are quite wide. About 25% of patients, and we see this not just in the HIV setting, but with all first-line TB drug reactions in SCAR, we often see about 25% of patients that will be reacting to one or more chemically distinct drugs. The commonest combination we see is rifampicin with ethambutol, but we've also seen, increasingly, strangely enough, rifampicin with pyrazinamide, and we're investigating whether there's some immunological basis for these dual sensitizations or these dual reactions. And then the other thing, and I mentioned this already, that we have gained experience from the clinical interface from, is that our patients seem to have a lower mortality rate for some of these severe reactions. And we are currently trying to devise a kind of case-controlled study where we can specifically tease out whether or not the fact that they're immunosuppressed is in fact some way mitigating and causing them to have a less severe immune-mediated reaction, which would be a reasonable hypothesis. Then the other experience we've gained is that even in some of the more severe reactions, and I showed you that in figure A in the previous slide, how you can actually treat through some of these reactions, and reinitiation or treatment through approaches have been successful. And we particularly found that in the case of efavirenz, which is a NNRTI drug that is now not really used anymore in our first-line regimens, but we found often would cause these DRESS reactions that could be safely treated through. And then I've already mentioned about the dual positivities. What's been encouraging back to the patient is that we've really found good long-term HIV/TB outcomes if a person is retained in care, and these outcomes look similar to what we would see in a patient, a general patient treated with TB. It illustrates, as I said before, there's still a high mortality related with this advanced HIV group, but if the patients are retained in care, you can see that their CD4 counts increase. Many of them have successfully completed their TB treatment, and certainly this is a very encouraging thing for our multidisciplinary clinic. But now, what about moving from the clinical insights to a little bit more immunology? Well, what these longitudinal times in hospital offer us, together with these sequential drug challenges, is multiple time points across a person's illness where we can get samples which might give us insights as to what immune cells are being activated by drug. And you can see here, this is one of the patients where we have been able to get multiple samples. We've been able to get good characterization of their phenotype. And then we've been able to conduct in vitro drug stimulation in addition to their in vivo drug re-challenge. If you look at the literature for these two types of severe cutaneous reactions, epidermal necrolysis on the left and DRESS on the right, they both involve the skin, and this is what this pictogram shows. And they involve predominantly T-cell mediated toxicity to the skin. And so I mentioned how in the case of epidermal necrolysis, these cytotoxic T-cells are attacking and destroying the keratinocytes. In the case of DRESS, there is more diversity in the infiltration with both CD4 and CD8 lymphocytes, and then also a slightly different profile of cytokines that are involved in the reaction. Increasingly, there are a lot of studies that are showing that resident tissue memory populations may be key as the triggering or inciting population being triggered by drug. And those T resident cells tend to carry markers like CD103 and CD69, and they seem to be showing some relationship to these reactions. In terms of our own work, and this goes back now to our couple of pivotal questions around drug hypersensitivity, what we've been able to show is firstly that, consistent with other data, we see these expanded cytotoxic T cells at the site of disease in our patients with epidermal necrolysis. This is single cell data that comes from 2 patients with nevirapine-induced epidermal necrolysis. Nevirapine is an antiretroviral NNRTI like efavirenz. And this reaction is also restricted by HLA to HLA-C0401 patients. And here what you can see on this map of the different cells that we find in the blisters. So when a patient is having epidermal necrolysis, you will often be able to access that blister fluid. And that blister fluid is particularly rich in these immune cells that are causing damage. And here you can see on this UMAP projection in blue, the big group is CD4 cells, and then particularly this green group, which is the cytotoxic CD8 cells. And if you look at it in comparison to normal skin and that skin that you find in a burn, like a traumatic burn blister fluid, you can see that in these immune-mediated reactions, there's a huge influx of these specialized cytotoxic drug-specific T cells. And in fact, together with our international collaborators, we're involved in putting now together a single cell atlas of the epidermal necrolysis. And what you can see consistently across these blister fluids of patients with epidermal necrolysis, irrespective really of HIV status or drug. And here you can see examples from allopurinol, from cotrimoxazole or sulfamethoxazole, trimethoprim, and also lamotrigine, is that you get an expansion of CD8 cells. But then if you look at the individual TCR repertoire, so within each group of CD8 cells or CD4 cells, you can look and do sequencing to look at the alpha beta pairings, what we call the CDR3 pairings of T cells. And what you see is that you get these oligoclonal expansions, and that's represented. So each little, and this is called a circus plot, and each green line represents one pairing of alpha beta, and the red indicates a single TCR clone. And so you can see across patients with allopurinol, sulfamethoxazole, lamotrigine, you're getting these expansions of oligoclonal T cells that are drug specific. If you look at their immune signatures in more detail, so you take those specific alpha beta pairings and you look at what genes they are expressing. You look in this right column here, these are from the 2 nevirapine patients. You see that they're expressing genes like granulysin, granzyme B, and these are cytotoxic markers, which are how CD8 cells do their killing of other cells. So you can see here that although the T cells differ in different reactions, so they're drug specific, and in many instances patient specific, they are still showing characteristic signatures. What's been interesting is in the context of HIV DRESS, some work done by one of our master's students, Tafadzwa Jembateta, has shown very nicely that although like in the literature, we see predominance of CD4 and CD8 cells as driving these reactions, in the context of HIV positive DRESS reactions in the skin, we see remarkably few CD4 cells. And this is, of course, consistent with what we see in the peripheral blood, which is CD4 depletion as you advance with HIV. However, interestingly, we don't see a very good correlation between what we see in the skin for CD4 number and what we see in the blood. And what's interesting here is that it speaks to the fact that although we have these different clinical entities of DRESS and epidermal necrolysis, there may be some overlap in the actual T cell that is mediating the reaction. And that insight comes from the fact that in HIV, we're just not seeing the same involvement of CD4 cells. Interestingly, there's actually clinically an entity we call DRESS-SJS overlap, and that is where you find features of both. So clearly from this data, both in HIV infected and unaffected patients, including from our drugs and our context, we see these cytotoxic oligoclonal T cells expanded and at the site of disease. So the next question that really we come to try and answer is how is these small molecule drugs, how are they actually causing an immune response in these affected T cells? Now, this is an area where there are several models and some quite substantial work in looking at different and supporting different models. But in essence, what we're looking at here is on an antigen presenting cells. If you are going to mount an adaptive immune response, you need to have an antigen presenting cell. And normally an antigen presenting cell will have on its antigen in the binding groove of what we call the MHC molecule or the HLA molecule, it will have a peptide. These peptides range in size between 8 and 12 peptides big. And then it will bind, if it's recognized by its specific alpha beta TCR pairing, okay, it's very antigen specific, this response, that will lead to a normal T cell activation response. This process we call signal 1 and is pivotal to developing any T cell response to any infection or other protein antigens. So one of the first mechanisms by which drugs are thought to provoke an immune response is by covalently binding to different peptides. An example of this that's been looked at is, for instance, penicillin allergy. The other would be the metabolite of sulfamethoxazole is thought to also bind proteins covalently. And what this does is effectively this creates a new peptide for the immune system to see and present. And that's what you see here, is that in the context of the presence of the drug or the metabolite binding covalently to the peptide, what you're getting is effectively a neoantigen that is being recognized by an immune response. However, this process here requires what we call antigen presentation and the complexing, because peptides are complex to HLA molecules inside the endoplasmic reticulum, so intracellularly. But sometimes to some drugs, we see that on the very first time you introduce the drug, often in a short space of time, you get an immune response. Now, that has led people to propose an alternate method, something we call the pharmacological interaction method. And there are various different types of this method, or permutations of this model, and includes from B all the way to F. And really what you see in this model is the drug, the small molecule drug, sitting at different spots in relationship to the HLA T-cell peptide combination or complex. And it's thought that in these spots, it's able to somehow stabilize or lead to binding and signaling through this complex. The third mechanism that probably has been most elegantly described is the method called the altered peptide repertoire, which has only been described in the context of the drug abacavir and abacavir hypersensitivity. What is being described there is that patients that develop abacavir hypersensitivity are restricted only to patients that carry HLA-B5701. So one single allele. If you carry HLA-B5703, you don't develop the reaction. And the crystal structure of abacavir with peptide in 5701 was solved. And what was very interesting is how, as is drawn in this diagram here, G, you can see that the drug was sitting actually in the binding pocket of the peptide. And what effectively it was doing was disrupting the conformation of the peptide. And so by doing so, it was making the repertoire of peptides appear completely new. So it was taking self-peptides and the sort of contents of your cell, the self-peptides within a cell are expressed at all times on class 1 MHC. And what it was effectively doing was in certain of these individuals that carry 5701, it was exposing their immune system to these new peptides. And so these are the sort of three methods. And when we've been thinking about our patients with TB drug reactions, we've been trying to determine which method may possibly best fit the reactions that we're seeing. So where have we started with this? We've started with this looking at rifampicin. And the reason we started looking at rifampicin is because in the laboratory, in vitro, we're able to get our patients having these reactions clinically in the clinic to show T-cell responses in the form of producing interferon gamma when we expose those cells to the drug in vitro. And this is what we call the interferon gamma ELISpot assay. And what this diagram shows is that we are able to, with rifampicin in patient with DRESS, show them developing significant T-cell responses to that. So what you can imagine now is we've got these patients that clinically we've exposed to the drug. We see that they're reacting, and then in the lab, we can now see that their T-cells are also recognizing the parent drug. So what that has enabled us to do is to then go and look at their genetics and see, do they carry a genetic risk for these reactions? And you can see from this table that we have found a putative B allele, which is a class 1 HLA allele in the form of B4403, okay? And this has been found in these patients in whom we see the rifampicin having a reaction in vivo. We have an ELISpot positive in the lab, and they safely get put on all their other drugs. Interestingly, we are seeing two different risk alleles across HIV positive and HIV negative patients. In one, we see HLA B4403, and in HIV negative, we see HLA B1501. These are compared to drug-tolerant controls, as well as, in many cases, to the South African general population from other studies of HLA. We've also looked at two other particular genes of interest, the first being the endoplasmic reticular aminopeptidase genes. These are genes which are responsible for the trimming of peptides in packaging to HLA, and then also the killer immunoglobulin receptors that are expressed to different degrees inhibitory or activating on natural killer cells. And natural killer cells have also been found at the site of disease. When we do some in silico docking of rifampicin, because rifampicin is quite a big drug, and we put it into the two putative risk HLA alleles, what we see is that in the context of HLA B4403, the best predicted binding for rifampicin is not actually in the place where peptide is interacting with MHC and potentially with T-cell receptor, but is actually at this intersecting place between the alpha chain and the beta-2 microglobulin. And this area here may be involved in the stability of HLA expression with peptide on the surface of cells. And you can imagine that the stability of HLA molecules and their peptides are very important just stochastically for their ability to activate the immune system. In contrast, if you look at 1501 and you try to in silico dock rifampicin, what you see is that actually, like one of those models we were showing in the previous slide, you'll see that rifampicin preferentially docks above the peptide directly on the area where you would find interaction with TCR. One of my PhD students, Puti Chosi, shown in this picture, she's imminently submitting her PhD work. And her PhD work has focused on taking that risk allele and then looking in the peripheral blood using high dimensionality approaches like time of flight mass cytometry or CITE-seq to see whether in these 3 patients with HLA-B44 and rifampicin reactions, whether we can see in the peripheral blood the particular offending populations of cells, like I showed you from some of that earlier CITE-seq data from nevirapin's SJS-TEN. And what we see there, if you look across the patients, and so you can see here what these bar graphs show. This is, if you look at the bottom here, the labels show one patient with different conditions. So pre-unstimulated, pre-RIF. What that refers to is it refers to a sample taken pre a drug rechallenge and then in vitro unstimulated or also stimulated with rifampicin, or a sample taken after in vivo drug challenge. And you can see here, there's a patient 1, patient 85, and patient 92. What you see here is, again, an expansion of CD8 and CD4 cells, but you don't see any clear pattern really easily across samples. But what you do notice is that if you compare pre-drug challenge samples to those when there's in vivo drug challenge on board, what you do is you see an expansion in the peripheral blood of CD8s, and you see a downregulation of monocytes and macrophages. Puti has done some amazing work trying to look across activation markers, across proliferation markers, to look at individual subsets of CD8 and CD4 populations to try and tease out what effector populations are reacting to drug. Unfortunately, we have not been able to clearly identify a drug-specific population that's consistent across these patients. And what is often difficult to dissect in this peripheral blood is that in HIV, what we are seeing is a strong effect of exhausted, expanded, homeostatically proliferated CD8 cells. We've had a little bit more luck if you look at the alpha-beta TCR pairings. And here you can see, again, those circus plots like I showed you earlier. This is looking at specific combinations. And this really picks out the dominant top 100 clones in the peripheral blood of TCR alpha-beta pairs. And what we've been able to do in looking and comparing different combinations is we've been able to find which particular TCR alpha-beta clone seems to be reacting to the drugs. So we look and examine both by comparing the pre and post drug stimulation in vivo and also pre and post in vitro. And you can see here that what we've highlighted in these gene panels here is in fact those similar genes we showed earlier in the form of granulysin and granzyme B. However, what we noticed, this is the data from one patient. If we compare across 3 patients, we in fact don't see a single same alpha-beta pairing across the patients. In other words, there's no public TCR that's the same amongst all these patients. What we've gone on, though, to do is to use a nice analysis called a GLP-H2 analysis, which what it does is it identifies sequences within different groups of TCRs that indicate that the peptide repertoires for those CDRs are likely to be very similar. So in other words, it finds these shared motifs that underpin and potentially underpin shared peptide repertoires. And if we use that analysis in our patients, what we see is we pick out only 8 motifs that you can find consistently across all the patients. And interestingly, if we look at this one motif, which goes by the sequence SQVP, we can again see that compared to other populations, when we stimulate with drug, we see expansion and upregulation of these cytotoxic and activation T cell markers. So this has given us some idea that, again, the CD8s are driving these stress reactions, that there is a drug-specific group of cells, and perhaps it's not the same across different patients, but that it shares some commonality in terms of the peptide repertoires that are found. And that would be consistent with the fact that it shows a similar HLA risk allele. But what needs to be done now is we need to take the single cell work and we need to put it at the site of disease. And unfortunately, our initial works on the skin biopsies to look for these alpha-beta pairings was unsuccessful, but we hope to move this forward. And then further, to clarify where exactly the rifampicin is docking on the HLA molecule, we need to do some more in vitro work. And we've actually teamed up here with chemistry, and we're going to be looking across different rifampicin analogs, because we have an interesting clinical observation that many of these patients reacting to rifampicin tolerate rifabutin. In the last few minutes of the talk, I'm going to focus on just one last aspect that is also fundamental and very important. And that is the issue that what in these patients, so we found the culprit T cell, we found the culprit place where the drug might be interacting with the immune system. But why do some people develop reactions and the majority of people remain immune tolerant? And in fact, what we call this is the positive predictive gap. And the reason for this is that most of these reactions, and I've shown you in the case of our rifampicin reaction that we are finding this risk HLA, but for many of them, there are other risk alleles that have been identified, drugs like carbamazepine, allopurinol, nevirapine. These all have HLA risk alleles that have been identified. However, the positive predictive value, in other words, the likelihood if you carry that HLA risk allele that you will in fact develop a reaction is often as low as 3 to 5%. And what that means is it means that the majority of people carrying that allele and being exposed to the drug, in fact, develop immune tolerance to this. There are several different lines of research here that are being explored. We've started to look across the ADME genes to look at drug pharmacology. Is there something related to the amount of exposure people have to a drug that impacts immune tolerance? It's logical to look at T regulatory cells, and I'll show you some of our early data in 1 or 2 slides. And then a lot of people are now looking at co-stimulation, because there's a lot of very interesting work that has come out of mouse models showing that in the case of abacavir, that as you deplete CD4 cells, you change the maturation phenotype of those antigen-presenting cells and their ability to activate an immune response that's coming through that T cell receptor. And so potentially what is happening is a change in those co-stimulatory signals that are coming through at the time that drug is being exposed to the immune system. Interestingly, in the context of our own patients, what we have seen is that several patients that have drug reactions to TB have had prior episodes of TB with no drug reactions. Then we've also had a patient in our registry that had a reaction to pyrazinamide, but subsequently, several months later, developed TB again. And when we reintroduced the drug to the patient tolerated successfully pyrazinamide. And the other thing, of course, is that several patients have had mild reactions where the drug can be continued and eventually the patient is tolerant to the drug. And what all of this has led us to believe is that tolerance is dynamic. And in fact, hypersensitivity may be something that even when you carry all of the genetic risks, might require these other tolerance mechanisms to be incorrect or to favor immune hypersensitivity rather than tolerance. And that might be dynamic and be able to be influenced and hence potentially predicted. We've put together these other ideas in the context of HIV, and it really relates to the last slide about how HIV specifically might be disrupting these immune tolerance mechanisms and making it more likely for people to react to drug. And then our starting off work, what we've noticed is we hypothesized just at the site of disease that maybe patients with DRESS in HIV have fewer regulatory T cells that infiltrate into their skin. In fact, what we found when we did the work was the opposite. So it does not seem to be the case that patients with these reactions in HIV DRESS have a reduced number of regulatory T cells. In fact, they have often expanded regulatory T cells. So if it is a problem related to T regulatory cells, it's a functional rather than a number problem that is occurring in the case of HIV. And this is work done as well by Tafadzwa. And then if you look further broadly in the literature, as I mentioned, how resident T cells are coming more and more to the fore, there are also other groups of antigen-presenting cells that have really not been studied. They've not been studied in the case of DRESS. They've not been studied in the case of HIV. And certainly this is something which warrants a lot of further work. And what Tafadzwa is now doing in his PhD is progressing from this initial immunohistochemistry and immunofluorescent approaches to looking in more detail using high dimensionality approaches like spatial transcriptomics and imaging mass cytometry. And what we are really doing is to shift our focus to look now at these tolerance mechanisms that might go awry, and to focus not just on the affected T cell populations, but to look at regulatory mechanisms and particularly at antigen presenting cells that might be the difference between a patient being tolerant or not. And similarly, what we are now going to do is, in the first part, we've kind of focused on looking at extreme phenotypes, but now actually shifting to look at patients that are tolerant, that become tolerant on drug exposure despite carrying the risk alleles. This might potentially give us even more valuable and generalizable insights beyond simply drugs. And of course, maybe at a high level to end off with, increasingly we are understanding that this broader concept of the exposome, okay, and if you look at our environment, our urban environment is constantly changing. We're facing climate change, air pollution, a barrage of different small molecules, chemicals, plastics, these things that are entering our environment. These are all impacting the immune system. And so undoubtedly drugs are just one kind of probe for the immune system. And certainly we are going to, in the coming decades and millennia, our immune system is going to face several more of these challenges and small molecule challenges from the urban and changing environment. And then, of course, as I mentioned in the slide around tolerance, these are not just new probes for hypersensitivity, but what the exposure might be doing is having crucial impacts on the dynamic control of immune tolerance. And potentially we are seeing some evidence epidemiologically that different autoimmune diseases are increasing. And this may also relate to similar changes from the exposome and influencing the dynamic mechanisms that are necessary for ongoing peripheral immune tolerance. But coming as an endpoint back to our patients, we of course hope to improve the ability to precisely identify these reactions using multi-omic approaches. And in so doing, the gold standard for these would be to screen patients prior to introducing a drug, and in so doing, prevent the terrible mortality and morbidity attached to these reactions. Thank you very much. And I've got several acknowledgements from different parts of Africa and overseas. Particular call out to my close colleagues, Ranks and Sipho Dlamini at our multidisciplinary clinic, and to my mentors, Elizabeth Phillip and Graham Menkes. Thank you very much.
Right. Thank you very much, Johnny, for the very exciting talk. I think we can all give a virtual hand of applause. I can give one that you can hear. And let's thank Johnny again for the very nice talk. So I think we can take a couple of quick questions for Johnny, please. You can show by raise of a hand, or you can type something in the text, and then I can read it off. Right, I am looking to
stand everybody. Stand everybody.
Yes. So there is a first question from Yanis. Please go ahead.
Thank you very much for a very interesting talk. Tell me, do you have an explanation for when individuals are hypersensitive to several drugs at the same time, drugs that belong to different classes? Do you think this is immunologically mediated, or do you have a different explanation?
Yeah, look, I think that is a very interesting question. We don't have a clear answer to that. We've got some clues. So for instance, there's some lovely work in the context of dual sensitization to vancomycin and cefepime that seems to carry, so vancomycin DRESS carries HLA 3201 as a risk allele. And interestingly, in patients duly sensitized they seem to have a different HLA. So there might be, as I showed you that figure of the way in which the drug is interacting with different components of the MHC TCR complex, it may be that certain positions can have multiple drugs interact and maybe lead to neoantigens and that might be the one mechanism. So there might be that would be truly what we call multiple drug hypersensitivity. The other mechanism that has been proposed, especially in the context of DRESS, where the immune system is already very activated. One of the proposals is that when you introduce a new drug, what you're actually getting is a non-specific flare-up reaction. And so rather than a true sensitization where if you take their T cells, you would see a drug-specific response, you're getting a clinical reaction without a necessarily lasting immunological T cell response. And that would be what we call a flare-up reaction related to immune reactivity. But I think the hard part is, of course, that oftentimes when you're seeing the patient, you don't know what they looked like before, whether they were originally sensitized. And in fact, my last point on that thing relates to what we're seeing with TB drugs. And I think because that's an interesting model, because there you're taking 4 drugs and introducing it at the same time. And so my hypothesis is that the dual sensitizations we see, so the multiple reactors, are in fact, like I was saying for vancomycin and cefepime, potentially their own group with their own way in which those 2 drugs are interacting with either the peptide MHC or, like I showed you in the models, a different position. And so they might be their own distinct group.
Thank you.
Right. Thank you very much. We can take one more question. You can show by raising your hand, then I'll give you the floor. Or maybe I can ask one quick question. So my take is, do you feel that some of this sensitivity to drugs could be improved? Does it require necessarily new drugs or to improve the way these ones are administered?
Sorry, just repeat that question again, Rip.
Yeah, my question is, do you feel that some of the sensitivity to the drugs could be reduced by having new therapies come on board, or by maybe improving the way the current regimes are being administered?
I guess that's 2 questions wrapped in one. So in the first one, I think there are definitely new drugs coming on board. And I think that, one of the questions we are considering for our next line of research would be actually to compare our sort of reintroduction approach and our model in terms of clinical care against simply switching these patients to a completely new TB regimen, because there are several now coming up by the WHO. So that's one thing we might consider, so we've developed this approach, but maybe the correct approach is actually just to completely avoid the drug and start an entirely new effective regimen with different drugs. Of course, we might face the problem that Eunice was discussing, where in the context of a reaction, we might get flare-ups or we might get other sensitizations. The second question, which is interesting, is whether the order of introduction of drugs may affect the sensitization pattern. That is something that we are trying to look at in our re-challenge data to see whether, if patients are re-challenged in a particular order, whether that has any influence on the reactions that they have. So that's a different question. I don't think necessarily, though, that that's clinically going to really make a difference, because practically patients that need to start TB treatment need to get more than one active drug at the same time. Otherwise, drug resistance will develop. So there's another force that's driving the way in which the drugs are being administered.
Okay, thank you. Any further questions?
May I ask one more question, please?
Yes, sure, please. Go ahead.
The concept of a hapten, I understood very well with antibody-mediated toxicities. Now, this is cell-mediated, and we know that the HLA binds, or rather the peptide binds to the HLA in the cell, in the reticular endoplasm. Am I correct? I hope I got that right.
No,
and then
100%.
And then it's expressed on the surface with the peptide. So do these drugs have to get into the cell to integrate with the MHC and the peptide, or do they bind on the outside?
Correct. So that's one of the key distinctions between the hapten model and the PI model. So in the hapten model, it needs to be packaged. So it needs that antigen presenting step through so the drug needs to get inside the cell and needs to be interacting with peptide MHC within the cell and within the endoplasmic reticulum, 100%. In the PI model, it's thought that the interaction with drug is occurring on the cell surface. Part of the reason, part of the kind of basic science work behind that comes from in vitro T-cell clone work, where what they've been able to do when they effectively clone out drug-specific T-cells, and then what they do is they block HLA, they do different kinds of washing steps, so they block antigen processing, so they can see whether, depending on how they impact the system, whether the ability of the T-cell activation remains. And that's how some of that work showing that, unlike in the hapten model, where we think definitely there's antigen processing required, there's some drugs that are interacting with T cells and MHC and peptide on the surface, and those can effectively, if you wash that away, you wash away the ability to cause an immune reaction. So yes, so it's thought that drugs can interact in different places.
So these self-peptides that are now expressed on the surface with the MHC ordinarily would not stimulate a cytotoxic T cell to bump off the cell, because these are actually peptides that are generally expressed on the surface. But do you think the then peptide converts it into something that is new, and therefore a T cell now sees it as foreign and then tries to bump off that cell? Is that how you think it?
Yeah, so well in the two models, like, which really not happening at the surface, the two models that come with antigen presentation, the one is that hapten model, and there effectively the haptenation to the peptide and drug, so that covalent interaction is effectively creating a neoantigen. That's a new antigen, the immune system is recognizing it. The altered peptide repertoire model with abacavir is abacavir is sitting inside the peptide binding pocket of the MHC, and it's altering the conformation of endogenous peptides. And there's some lovely work where essentially the C terminus of the peptides are changed by the presence or absence of drug. And so effectively there, the presence of the drug is creating it's turning endogenous peptides, which are tolerant to the immune system in certain individuals, into a new set of, again, neoantigens that the immune system is recognizing as foreign.
Got you. Thank you very much.
Right. Thank you very much. I think this time we can now bring this session to a close. And let's thank Johnny again for a very fantastic talk and for the stimulating discussion that has taken place afterwards. So Johnny, thank you very much. And we look forward to having all of you again for the next series that will be on the 16th of August, when we have another meeting of the Royal Society. Thank you, everyone.
Very much, Ruben. Thank you for having me. Have a good evening, everyone.
Right. Thank you. Right. Yeah. Thank you, everyone. I think we can bring it to a close.